Now, moving on to our next subtopic, which is interconnected to what we just studied, that is ribosomes. Now, what exactly are ribosomes? Ribosomes are small, dense, rounded, and granular particles of ribonucleic acid. Okay? Now, let's have a look of the structure of ribosome. Now, you can see I have drawn two circular bodies. One is bigger in size, right? And the other one is smaller in size. But very clearly, we can see that it is made up of two circles joining together. ठीक है? Sorry. So basically, हमारे पास ribosomes is made up of two subunits. One is a large subunit and the other one is a small subunit. Do they have label names? No, they are called large subunit and small subunit. They do not have any specific names. Okay. Now, what is a ribosome made up of? Ribosome is made up of two things only. It consists of protein and ribosomal RNA. ठीक है? So it has 35% protein and 65% ribosomal RNA. You do not have to know these percentages, these values, just for your understanding. I've added over here. It's not part of your syllabus. You just have to know that ribosome is made up of protein and rRNA. And now notice, हमारे पास ribosomes कहाँ पर बन रहे थे? हमारे पास ribosomes बन रहे थे inside our nucleolus. Nucleolus was responsible for manufacturing ribosomes. Is it the same protein made from protein synthesis from nucleus? Okay, so Huda, in nucleus, I said that we are only making the mRNA. The process of protein synthesis is not complete. In nucleus, there is only one step, one part of protein synthesis which is occurring, which is basically the formation of mRNA. So we can say that we have protein synthesis. And how does protein synthesis occur? Protein synthesis occur in this manner. We have DNA, basically. Then that DNA is formed, is transcribed into an mRNA. Okay? And only this process, this transcription process occurs inside the nucleus. So only this part is occurring inside the nucleus. So we are yet to form the polypeptide and the protein. The mRNA would exit the nucleus and the remaining process, the second part, which is translation, that will occur in the cytoplasm. So in the nucleus, we have only formed mRNA, and then the nucleolus had formed the ribosomes. And ribosomes, as we just studied, is made up of ribosomal RNA. And it comprises of 65% ribosomal RNA. Now, obviously, ये ribosomal RNA, because it was being formed in the nucleolus, so this nucleolus possesses the ribosomal RNA, right? So we can say, so where do the proteins come from? But the protein basically do not come from anything. We form the protein in a step, in a mechanism called the protein synthesis mechanism, which starts from DNA, a gene. Okay? One particular gene is specific for a polypeptide, right? For example, let's take the example of an amino acid. What do you say? Let's take the example of tryptophan. You do not have to know this name. I've just named an amino acid. Now, for example, gene T is responsible for coding tryptophan. ठीक है? उसी तरह से gene A is responsible for coding something like glycine. Okay? Now, every particular gene is responsible for coding one amino acid, and all these amino acids then combine together to form a polypeptide, and then these polypeptides join together to form protein. So technically, we are forming a protein from a gene. So gene is basically the starting point of protein synthesis, okay? And the first step of protein synthesis, which is transcription, formation of DNA into mRNA, that is occurring inside the nucleus. So within the nucleus, हमारे पास जो end product है, that is mRNA, okay? Is this clear, Huda? Now, हमारे पास we just understood that ribosomes are made up of two subunits. There is a large subunit and there is a small subunit. They are made up of protein and ribosomal RNA. Now, it's very important to understand that they're not surrounded by a membrane. Now, let's take a pause over here and go back to our previous lecture in which we said that prokaryotes are those organisms, those cells which do not possess a true nucleus and which do not have any membrane-bound organelles, right? Now, ribosomes are not surrounded by a membrane. They are not membrane-bound. So, do you think that prokaryotes would be having ribosomes or they would not be having ribosomes? What do you guys think? I want you all to answer this question. Should prokaryotes possess ribosomes or they should not possess ribosomes? Okay, so Muskan is saying that yes, prokaryotes should have ribosomes, and so is Huda saying. You guys are right, because ribosomes do not possess, are not surrounded by a membrane, so they would be present inside a prokaryotic cell. ठीक है? Because it is not a membrane-bound organelle. Now there are two types of ribosomes. ठीक है? One is a 70S ribosomes and the other is an 80S ribosomes. Now, the type of ribosome which is found inside a prokaryotic cell are the 70S ribosomes. Ashra, now what is the difference between a 70S ribosome and an 80S ribosome? The only difference is the difference in size. ठीक है? 70S ribosomes are smaller in size, 80S ribosomes are larger. ठीक है? That's the only difference that 70S ribosomes and 80S ribosomes have, which you should know at your level. So 70S ribosomes are found in prokaryotic cells, they're found in mitochondria, and they're found in chloroplast. ठीक है? So there are three places where we can find 70S ribosomes, which are prokaryotic cells, mitochondria, and chloroplast. Now, where do we find 80S ribosomes? Now, 80S ribosomes are found in eukaryotic cell. Yesterday, when we were studying animal cell, we said that हमारे पास rough endoplasmic reticulum is that endoplasmic reticulum which has ribosomes on its surface, which in turn gives it a very rough structure, a very rough look. So now we have ribosomes present in eukaryotic cells because they are present in RER, and they're not only present or attached onto the RER, they're also freely suspended in the cytoplasm. यानी कि हमारे पास ribosomes दो जगह पर होती हैं. हमारे पास ribosomes, they're attached to RER as well as they're freely suspended in the cytoplasm. But 80S ribosomes are only found in eukaryotic cells, which means that they would be present in prototists, in fungi, in animal cells, and in plant cells. They would not be present in a bacterial cell because we know that bacteria belong to the kingdom Prokaryota. Okay? So, ADS ribosomes are only present in eukaryotic cells, which means that on the RER, if we have a ribosome, that is an ATS ribosome, and if we know that it is a eukaryotic cell, which means any cell amongst these four species, and it is found in the cytoplasm of those cells, then it would be an ATS ribosome. Otherwise, if we see a ribosome in a prokaryotic cell, in a mitochondria, or in a chloroplast, it would be a 70S ribosome. Is it clear so far? Okay. So now, as Uda was asking us, where does translation occur? So ribosome is that place, it is that site where translation occurs. So what happens is that, let me draw over here. So we have a nucleus. This is our nucleus. ठीक है? Let me just draw some nuclear pores. These are my nuclear pores. ठीक है? Now, for example, now I want you guys to answer me. Which process would be occurring inside the nucleus? Which process of protein synthesis would be occurring inside the nucleus, and what product would be formed inside the nucleus? I want you all to answer in the chat box. Yes, Muskan, you are right. And what is the product of transcription? End product क्या बन रहा होता है transcription का? Yes, very good. So we have transcription happening, and transcription is what? That the DNA inside the nucleus is converted into what? It is converted into mRNA. This is what we call transcription. So transcription is our first step of protein synthesis, which occurs inside the nucleus. Now this mRNA, because we know that DNA cannot leave. Acha, one more thing that I forgot to mention. Now, gene, when I was explaining this to Huda, that the protein is basically coming from a gene. One gene is responsible to code for a particular amino acid, ठीक है? For a particular polypeptide, sorry. One gene is forming a polypeptide, and then multiple polypeptide genes are connecting, coming together to form protein. So technically, if I go back over here, I can easily say that my gene is basically the start point from which my protein is being made, right? I can easily say that if I have to tell where is the protein coming from, I would say protein is coming from a gene. DNA is coding for a protein, right? That is basically the origin of a protein. But the problem that we are facing, just ki wajah se we are having these two steps, is that DNA cannot leave your nucleus. And protein cannot be formed inside the DNA because the nucleus does not have that much capacity, that much space. So what happens is we need a messenger, we need a mediator. इसका बहुत सिंपल सी बात हुई. Let's take an example of WhatsApp. WhatsApp क्या करता है? I have to message, for example, I have to send a message to Huda, right? Now I am the person who's sending the message, so I am the start point and Huda is my end point. But obviously मैं एक जगह पे रहती हूँ, Huda एक जगह पे रहती है, so I cannot communicate to Huda without a mediator, without a messenger. So I need something, right? For a DNA to form a protein or to form a polypeptide, that cannot happen directly. Why? Because DNA अपनी जगह नहीं छोड़ सकता और polypeptide या amino acid nucleus के अंदर नहीं आ सकता. So we need a mediator, we need a messenger, and that messenger over here is our mRNA, right? So this entire process is occurring because of the fact that DNA cannot leave the nucleus and amino acids cannot come inside the nucleus to join together to form a polypeptide. So because we need a mediator, we are having this process of transcription, which is forming an mRNA. Now this mRNA is basically carrying the message कि अच्छा इस वाली जीन ने ये mRNA बनाया है, तो ये mRNA अगर मुझे हुदा को भेजना है तो ये हुदा के पास जाएगा, अगर मुझे हाजरा को भेजना है तो ये हाजरा के पास जाएगा, अगर मुझे मुस्कान को भेजना है तो ये मुस्कान के पास जाएगा. So every single mRNA, every single message is specific to a specific polypeptide. ठीक है? So ऐसा नहीं है कि कोई भी जीन कोई भी पॉलीपेप्टाइड बना देगी. No, every gene has its own code, every gene has its own message, and that mRNA carries that message of the DNA. And mRNA would always ensure that it will form the specific protein which the particular gene had to code for. Okay? Any questions so far? Okay. So basically now this mRNA leaves the nucleus through our nuclear pore. Now we have this mRNA out in the cell. ठीक है, ये हमारे पास cell का cytoplasm. Now this mRNA would see ribosome. ये मेरे पास ribosome है. Let me draw ribosome. And it will go and attach itself to this ribosome. And now this ribosome, which has an mRNA on it, would attach a further few things. हमारे पास tRNA amino acids लेकर आएगा, पूरी हमारे पास polypeptide chain बनेगी, amino acids से polypeptide से finally amino acids basically will join together to form a polypeptide. But basically long story short, हमारे पास mRNA आकर attach किस पर हो रहा है? राइबोसोम पर हो रहा है. और फिर ये mRNA, let me just write that this is, ये mRNA यहाँ आकर attach हो गया इससे, ठीक है? This mRNA then will be converted into the polypeptide. ये पूरा एक process है which we will be studying in chapter 6. तब हम इसकी detail में जाएंगे. But this is basically my translation where my mRNA is translated into a polypeptide. ठीक है? This process is occurring on the side of a ribosome and therefore ribosomes are responsible for translation. Any questions so far? So basically, so far we've done nucleus and we've done ribosomes, and we understood that nucleus is basically the site of transcription and ribosome is the site of translation. So basically, nucleus and ribosome are basically the two places where our protein synthesis is occurring. Now, हमारे पास ribosomes we understood या तो cytoplasm में freely suspended होते हैं, they are present within the cytoplasm, freely suspended, or they're bound to the rough endoplasmic reticulum. So our mRNA can go and attach to the ribosome freely suspended in the cytoplasm, or it can also attach to the ribosome present on the rough endoplasmic reticulum. क्योंकि हमने अभी पढ़ा कि हमारे पास यूकेरियोटिक सेल में ATS राइबोसोम्स होते हैं, और वो राइबोसोम्स या तो हमारे पास RER पे अटैच्ड होते हैं या फिर वो हमारे पास फ्रीली सस्पेंडेड इन द साइटोप्लाज्म होते हैं. तो यूकेरियोटिक सेल्स में जो राइबोसोम्स होते हैं वो एक ही जैसे होते हैं. So this mRNA can go and attach to the ribosome of the RER, or it can go and attach to the freely suspended ribosome, and the process would be the same. ठीक है? Any questions so far? Okay, so I have a question for you guys. If ribosomes are attached on the surface of rough endoplasmic reticulum and mRNA can go and attach to the ribosomes of the RER, can protein synthesis occur on the surface of rough endoplasmic reticulum, or can it not occur on the surface of rough endoplasmic reticulum? What do you think? Can RER be the site of protein synthesis in the form of translation or not? Okay, so I have Huda's answer. What do you guys, the rest of you, think? Yes. So because the ribosomes that are present on the surface of rough endoplasmic reticulum can easily allow the mRNA to attach to itself. So we can say that one of the functions of rough endoplasmic reticulum is that it is involved in protein synthesis via translation. Why? Because of the presence of ribosomes. I'll just make this note for you guys over here. Because of ribosomes. Now, chalo, एक function we have understood of RER. Let's just understand what rough endoplasmic reticulum itself is. Now, even in the diagram that I showed to you guys yesterday of the annual cell, and even in this diagram, there is one thing that you can see, which is that our rough endoplasmic reticulum is very closely attached. ये हमारा rough endoplasmic reticulum यहाँ से शुरू हो गया है, और ये हमारे पास बिल्कुल purple में we have our nuclear membrane. So usually, not always, but usually most of the time, our rough endoplasmic reticulum is continuous with the nuclear membrane. Now, why do you guys think that rough endoplasmic reticulum is continuous with the nuclear membrane? Smooth endoplasmic reticulum की तरह freely suspended या कहीं पर भी वो क्यों नहीं exist कर रहा cytoplasm में? Why is it specifically continuous with nuclear membrane most of the times? Any idea, any suggestions, any guesses? Is related to what we studied just now. So try to connect what we understood, that the entire process of translation and transcription. How can we connect this with the fact that rough endoplasmic reticulum is usually continuous with the nucleus of the cell? Yes, very good, Muskan. So basically, the mRNA, let me draw this for you. So basically, we have our nuclear pores over here. Now our mRNA is leaving like this, right? So it is not like that mRNA cannot reach to, it can reach to the freely suspended ribosomes as well. And even if our RER is freely suspended in the cytoplasm, the mRNA can easily travel to it, attach to it, and undergo the process of translation. But it is just easy. For example, a university hai, for example, you have a choice to choose between two colleges. One college is near to your house, the other college is far away to your house. And both the colleges are offering the same facilities, both the colleges are offering the same both the colleges are offering the same faculty, the same opportunities, the same subjects to you, the same fee structure. Everything between those colleges is same. The only difference is that one of the colleges is nearer to where you live and the other one is far away from where you live. So obviously you would prefer the college which is near to your place because it would be easy for you to reach. Same is the case over here. The mRNA can reach this rough endoplasmic reticulum if it is at a distance. It can also reach to this freely suspended ribosomes, but it's just easier कि जैसे ही वो nuclear pore से निकले, it interacts with the ribosomes on the rough endoplasmic reticulum and undergo translation and form a polypeptide. ठीक है, that is why it is usually, not always, but most of the time, continuous with the nuclear membrane. Okay, so Huda is asking, it happens on both RER and SER. Okay, Huda. So you guys said to me that because ribosomes are present on the rough endoplasmic reticulum, that is why translation can occur on rough endoplasmic reticulum. Does smooth endoplasmic reticulum possess ribosomes? Are there any ribosomes present on smooth endoplasmic reticulum? Okay, so you are saying ribosomes are present on smooth endoplasmic reticulum. Okay. So remember yesterday, I said to you to remember this way about RER. I said that endoplasmic reticulum, which has ribosomes, is basically the rough endoplasmic reticulum. And I said, remember it this way that ribosome starts with R and rough endoplasmic reticulum starts with R. So this is common between both of them. So any endoplasmic reticulum which possesses ribosomes on its surface is a rough endoplasmic reticulum. So the rough endoplasmic reticulum consists of ribosomes, these small ball-like structures, ये इसकी ribosomes हैं, ठीक है? And because of this, when you are observing rough endoplasmic reticulum under a microscope, you would see that because of these grains, because of these ball-like structure, which we know are ribosomes present on its surface, a very rough, a very bhurbura sa, I don't know if you can understand what bhurbura is, but a very rough, a very grainy, you can say, structure is appearing under the microscope side. On the contrary, we have a smooth endoplasmic reticulum. It does not have any granular ribosomes attached onto it. It is a very smooth structure, a very smooth membranous structure, and therefore, because smooth endoplasmic reticulum does not have ribosomes on it, it does not have a rough structure. It does not have a grainy structure, a grainy or rough appearance, and because a smooth endoplasmic reticulum does not have ribosomes, it will not undergo translation, which is your protein synthesis. Does that answer your query, Huda? Any questions about it so far? Okay, perfect. Now, so as I just mentioned, surface of RER contains ATS ribosomes, thus giving it a rough appearance when observed. Okay. Now, if someone asks you that describe or define a rough endoplasmic reticulum, how would you do that? So basically, endoplasmic reticulum, be it a smooth endoplasmic reticulum or be it a rough endoplasmic reticulum, you have both rough endoplasmic reticulum and smooth endoplasmic reticulum in this picture. So let's see if this statement answers for both of them or not. An endoplasmic reticulum is a system of interconnected, flattened. The first word is system of interconnected. That is our first description. Flattened, okay, membranous sacs. These are our three key words to describe rough endoplasmic reticulum. Let's understand why we are saying. Now, if you could notice, I'm gonna outline this for you all. Just give me a second. Okay. Now let's outline our rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, if you could see my outline. Now, do you notice that these things are interconnected to one another? Can you notice this? Can you appreciate this? That ये एक layer है, ये दूसरी layer है, ये तीसरी layer है. There are different layers, but they are interconnected to one another. Can you notice this? Is it noticeable, observable to you that how they are connected to one another and not separate? Subsequently, if I draw the same for the rough endoplasmic reticulum, let's draw for them. Let's have a look. So you see these layers are all interconnected to one another. So first and foremost, it's a system of interconnected. We've just visualized how they are interconnected to one another. And then it goes on to say flattened. Now, if you could notice, they are very flat. It's like if you take something which is very airy and you put a very and you put your hand with a lot of force on it and it becomes flat, right? So their appearance is very flat. It's not a very puffy, a very airy appearance. It's basically, it is a 3D structure. We can see that there is some space, some compartment within them, but it's very flat. It's not very airy, or it's not very full. It's a very flat appearance that we can see. So these are basically membranes. Basically, your endoplasmic reticulum, your smooth endoplasmic reticulum, rough endoplasmic reticulum, these are basically मेम्ब्रेनस सैक्स, यानी आपकी बहुत सारी मेम्ब्रेन की लेयर्स को हमने इंटरकनेक्ट करके फ्लैटन कर दिया है, एंड वी हैव फॉर्म्ड बेसिकली अ स्ट्रक्चर. एंड these membranous sacs, because obviously it's not one membrane, ऐसा नहीं है कि एक membrane से बड़ा है, it's a lot of membranes. Let me even draw it for you. So basically something like this in rough. So these are a lot of membranes. For example, this is one membrane, second, third, fourth, fifth. So these are a lot of membranes which are interconnected and flattened to give you this appearance. Now, what are these membranous sacs called? So they are basically called, there is a name for them, which is cisternae. They are called, collectively, they are called cisternae. Cisternae is a plural. One membrane sac or one membrane layer is basically called cisterna. This is singular and this is plural. Yani collectively, if I have to call them, I would say that an endoplasmic reticulum is a system of interconnected flattened cisternae. And if I'm talking about one particular membrane, then it is a cisterna. Okay. Any questions so far?